All right, I'd like to introduce our next speaker, Charlotte Thompson from ProImmune. Charlotte is one of ProImmune's top immunology sales specialists based in Oxford and representing the company in the European region. Charlotte graduated from the University of Aberdeen, where she studied pharmacology before joining Novo Nordisk as a member of their field team, specializing in their GLP-1 agonist portfolio. Charlotte has been with ProImmune since joining in January of 2022 and is responsible for supporting our customers from inception of a technical study design through to completion. And the title of the talk is Ankyrons, the Future of Target Binding Reagents Beyond Antibodies.
Hello, everyone. Thanks, Emily, for the introduction. And thank you again to everyone for being here and taking the time out of your day to attend and hear the insights shared by our speakers. So earlier in the conference, Emily and Edmund already introduced ProImmune and some of our capabilities. And today I'm going to be talking to you about our newest technology called Ankyrons. So Ankyros are next generation target binding reagents that go beyond antibodies. This might seem like quite a bold statement. Antibodies have been the mainstay of kind of target binding technology and research for such a long time. But as you'll come to find out, these proteins have some pretty unique properties that we believe make them superior to antibodies in the research space. So just to sort of paint a picture, I'm sure many of you will have experienced considerable frustration around trying to identify antibodies against a target of interest. If you then choose to pursue the options of custom antibody generation, you soon come up against the significant kind of current constraints of this approach. It is a high-risk, expensive, and time-consuming adventure. Especially now with the exponential increase of transcriptomics data driven through technologies such as RNA-seq, and the clear sort of need to investigate a vast array of novel proteins, the pedestrian nature of antibody generation has become a severely limiting factor. So imagine now if instead you were able to generate these target binding reagents to not just one or two proteins at a time, but all proteins within an entire pathway, or your entire pipeline of therapeutics, or even against all the proteins expressed by a specific virus. So this technology now enables what was previously unimaginable to become a reality in a rapid and cost-effective manner. Ooh. See if I can move off the foot. There we go. So Ankyrons are based on single domain Ankyron repeat scaffold proteins, and they are essentially small looped proteins. They've already been really well characterized in the clinic, but never before applied to the research space. So essentially the reverse of what happened with antibodies. And these Ankyron repeat scaffolds have a really high diversity, which means that we've generated a Terra library of over 1 trillion clones to date. We can then use this Terra library to pick out specific binders against a target of choice in a single pass using ribosomal display. This is unlike other approaches such as phage display, which typically requires multiple rounds of screening and affinity maturation steps. And as you can sort of see here, compared to a typical antibody, they're about 1/10 of the size, which means you can get some really nice high resolution images, and it also makes them perfect for intracellular staining as they can penetrate your cells with ease. Ankyrons are generated using completely animal-free methods and therefore by default are recombinant monoclonal sequences. Because they're synthetic molecules, they're also not constrained by immunological tolerance, and so can be targeted against all proteins, even self-proteins. You also don't have to consider any isotypes to consider, simply because they're just not antibody molecules. Rather, they are small, kind of single domain proteins. They're highly stable. They're highly heat stable too, so we ship these at room temperature, and they can be easily formatted depending on your desired application. In terms of application, Ankyrons can be used in anything that you would typically use an antibody for. So that can include flow, ELISA, immunohistochemistry, and many, many more. Currently, we have a catalogue of around kind of 1,300 specificities, and this is rapidly expanding as we dive into new research areas. So all of our catalog Ankyrons typically ship in about 2 to 3 weeks, and we send you as many clones as we have available, up to 6 at a time, against your target. most Ankyrons, sorry, have multiple clones for you to test. And all of these clones have been confirmed as binders using a sensitive immunoassay. And most have a signal to noise. They must have a signal to noise ratio of greater than 10 to 1. Most are in the hundreds of thousands, and some even reaching kind of millions to 1. We provide these clones as standard with a V5 tag in the first instance, meaning that you can test the multiple clones and pick out your preferred clone, which can then be directly labeled with pretty much any label that you could ever require. And now, whilst having the growing catalogue of off-the-shelf reagents is, of course, extremely useful, the technology really comes into its own when you start to think about generating these reagents in a custom manner. So as I sort of touched on earlier, Ankyrons are more cost-effective than traditional antibodies in every way. And that owes to a few different things, including our high-yield expression system, the efficiency of the ribosomal display technique, and our investment into high-throughput systems. This therefore opens the door to progress research ideas that typically would just not have been, feasible by generating custom antibodies. For example, for proteins found in veterinary or plant science. And we can generate these custom Ankyrons in approximately 8 weeks from receipt of target protein. Of course, as we found over the past couple of years developing this technology and working with collaborators, not everyone has that protein to hand. So off the back of this, we've developed our own protein expression system, specifically for the synthesis of target proteins to be used for Ankyron discovery. This means that the only input we need from you is the kind of ID of your target protein. We can then go ahead and express the protein or a domain of the protein in-house, stick it straight into the Ankyron screening, and the end result being multiple custom clones for you to test at a fraction of the cost and time that it would have taken you to generate a completely custom antibody. And these Ankyrons can also be formatted in pretty much any way you like. We can make them bi or multi-specific. We can even daisy chain Ankyrons of the same specificities together to increase avidity to the target. And whilst this is an option, it's certainly not necessary, as each Ankyron binder has been automatically selected to be of either medium to high affinity only. So we're talking kind of nanomolar to picomolar affinities here. And depending on your application, you might not actually want a really high affinity clone. And we can determine this in-house using a solution equilibrium titration assay to select the best clone for your application. So there's an example of some of this data on the right here. Ankyrons A1, 2, and 3 are all confirmed binders to the same target, PDL1, but we're all determined to have different affinities using a solution equilibrium technique. And they range from 600 picomolar, so on the right, the kind of purple line, down to 0.7 picomolar at the red line on the left. As well as determining their affinity, we can also distinguish closely related proteins using a guided selection technique. So with both proteins in hand, we can generate and select for those that will bind to protein 1, but then deselect those that also bind to protein 2. And this can be performed down to a really specific level, including peptide MHC complexes. And that brings me nicely to my next slide, and that is how to date we have had success generating these Ankyrons against targets that have been notoriously challenging in the past to generate using the kind of typical antibody approach. So if we think about the therapeutic arena, obviously there is a huge interest in anti-peptide MHC antibodies or TCR-like antibodies. And these are quite challenging and also very costly for groups to make. For example, if you want to immunize a mouse population, you're going to need a large quantity of MHC monomers to immunize your mouse and then hopefully generate antibodies. However, using Ankyrons, this is actually quite simple. And we already have multiple Ankyrons against peptide MHC complexes in our catalog. Some of these are listed to the side here, and these include kind of cancer and viral specificities too. So we've used the guided selection process to ensure that the Ankyrons are specific for the complex as a whole. So not just the peptide or the MHC, and not for closely related peptide MHC either. And some nice data to kind of show this was kindly provided from one of our collaborators at Duke and US, Professor Bertoletti, and they are working with HPV. So we made an anti-HPV MHC peptide complex Ankyron, and they used those to stain an HPV cell line that had been pulsed with interferon gamma. And this stain here is shown in green. The blue histogram is where they stained those same cells, but using Ankyrons raised against an irrelevant peptide, but restricted to the same HLA type. So what you kind of see here is some really nice exquisite staining to just the MHC complex of interest. And then down below, another really important kind of area of interest at the minute in therapeutics are, of course, anti-idiotype antibodies. But again, these are generally quite time-consuming and expensive to make. We have made multiple Ankyrons already on our catalogs against a range of these biotherapeutics. So this is an example of some anti-Avastin Ankyrons binding only to Avastin, not to any of the other therapeutics in the panel, and also not to the IgG isotypes. Now, next here we have some data from Dr. Jacqueline Smuckley's lab at the University of Bern in Switzerland. And this is a great example of where we've provided a reagent where previously one just did not exist. So this group is interested in Plasmodium burgeri. This is the causative protein for rodent malaria. So their target protein is GABARAP, which has a role in cellular transport. However, Jacqueline kind of contacted us and explained that even though they tried, sourcing the antibody for this GABARAP protein was extremely difficult. As it happened, we actually had multiple GABARAP clones already in our Ankyron catalog. So we sent them to her for testing, and they used them in some immunofluorescence staining shown here. So they infected wild-type HeLa cells with Plasmodium burgeri for either 6 or 24 hours. And then they visualized the parasite in the vacuole membrane of the host cell. So this is shown in the far left column here. In the second column, you see the staining with the GABARAP-specific Ankyrons. And for the first time, they were really able to show what they thought to be true, which was co-localization of GABARAP and the parasite. And that staining was also shown to be specific. As you can see in the kind of bottom 2 rows here, they then went on to use a triple knockout HeLa cell line, and we see kind of no staining with those Ankyrons around the vacuole. And this group is planning on publishing with this data very shortly too. So in some instances, you might, the reagents might exist, but they just don't work great for your application. And this was an example of where there simply weren't any really good antibodies against hypoxia-inducible factor 1 alpha or HIF1 alpha, which is a transcription factor. So we made anti-HIF1 alpha Ankyrons for Professor Wither's lab in Birmingham. And they used these in some flow staining with a black 6 mouse model where they injected MC38 tumors. They then isolated the NK cells within the tumor and spleen and use the Ankyrons to stain intracellularly. So we see for 3 different mice represented by the 3 different histograms here, you can see upregulation of both the splenic and the tumor NK cells compared to the control, specifically showing elevated levels of NK cells in the tumors, also shown in the MFR bars on the right. And I think it's important to point out that this data, as well as all the other data that I'm going to be showing today, was essentially generated as a first pass on receipt of the Ankyrons. So none of this data was following months and months of optimization like you might typically have to do with an antibody. So much like antibodies, Ankyrons can have different characteristics, whether that be to block, neutralize, or act agonistically. And you'll see a couple examples of this over the next few slides. So in this collaboration, we generated Ankyrons against the SARS-CoV-2 spike protein. And we did that for the wild type virus as well as the Delta and the Omicron mutants. So these collaborators used biolayer interferometry, which is a label-free biosensing application. And this measures association and dissociation. In the upper 2 lines, the green and the red here, you see where the wild spike protein is in close association or binding with Ankyron clone 41041, and that has been raised against the wild type spike. Then down below these bars here, you can see that the clone does not bind when incubated with a MERS spike, and neither do the Delta or the Omicron clones, or the other wild-type clones, showing it is specific to SARS-CoV-2. And this is just an example of an either higher affinity wild-type Ankyron clone that is noted by no dissociation or a flat line on top. And then on the bottom here, they use the wild-type spike protein again, but incubated with irrelevant Ankyrons or Ankyrons that had been raised against the Fc gamma receptor. So again, no cross-reactivity was detected. I'm going to try and fly through these next slides in the interest of time. Next, we have some affinity binding graphs. So we have kind of high affinity Ankyron clones against the wild type spike protein, as well as the Delta and the Omicron mutants. And again, these have been selected for in a single pass. So the X and the Y axis are the on and the off rates, and then the diagonal lines represent the affinity. And each different color represents a different Ankyron clone. There are replicates within those clones too. So the highest affinity binders are shown here in the kind of upper left corner of each figure. For example, one of the wild type clones had an affinity in the 90 picomolar range. And finally, from this set of collaborators' data, they also then wanted to look at the ability of those Ankyrons to neutralize the virus. So using pseudoviruses in combination with either the Delta or the Omicron Ankyron clones. And then they measured the percentage neutralization. So on the left here with the Delta Ankyrons, you can see that they did induce 100% neutralization, which was essentially diminished out as you kind of dilute the Ankyrons. But on the right with the Omicron Ankyrons, you can see a much more shallow curve, and they maintain a much higher percentage of neutralization, which is consistent with the fact that they have much lower IC50 values. They were therefore able to demonstrate that the Omicron Ankyrons were of a really high affinity and also possess neutralization capacity. So just to end on, we have some internal data that was generated by our lovely lab team here. And this was generated using a HeLa cell line. As I'm sure you're aware, cells obviously contain various membrane-bound organelles, and they each contain specific proteins related to their function. So we have a panel of Ankyrons that detect these specific organelle proteins, and then allow you to therefore identify the subcellular location of your protein of interest and investigate the role that it may play within the cell. So at the top here, you can see some epifluorescence microscopy images of Ankyrons that recognize the chaperone proteins calreticulin and calnexin, and they are located in the endoplasmic reticulum. So the endoplasmic reticulum is obviously involved in the synthesis of proteins, lipids, steroids, and is continuous with the outer membrane of the nuclear envelope. So it's apparent in close proximity with the nucleus. This image and the rest of the images shown have been detected with an APC conjugated secondary antibody that then recognizes the V5 tag on the Ankyrons. So that's the red staining. And then they've also been counterstained with the DAPI shown in blue. Below and to the left is our kind of Golgi apparatus marker. Ankyrons bind RCAS1, which is a protein that regulates vesicle formation, secretion, and protein glycosylation. And here we've labeled the HeLa cells with an RCAS1 binding Ankyron clone, where the specific staining has allowed us to visualize the Golgi apparatus to a really high resolution. To the right, there's lysosome staining, using Ankyrons against tetraspanin CD63, which is a protein found in late endosomes, lysosomes, and secretory vesicles within the cell and in the perinuclear compartments. And finally, here you can see staining using Ankyron 00521, specific for the cell surface adhesion receptor CD44. And CD44 is an almost ubiquitously expressed protein, which is found on the cell surface of epithelial, mesenchymal, and hematopoietic origin. It's also worth noting that all of these Ankyrons shown so far are effective at labeling organelles in both unfixed and formaldehyde fixed cells. This means that if you're a group that has capabilities to perform kind of live cell imaging, then these could be really handy for you. And our final epifluorescence image here was generated using Ankyrons against histone H31, which, as you can see, specifically stain the nuclei and co-localize with the DAPI stain shown here in blue. So just to finish off, now you've seen kind of several examples of how we can raise Ankyrons against essentially any structural protein, yielding access to new proteomic areas and disease states or different species that just weren't accessible previously. And we can do so in a very cost-effective and timely manner, which will accelerate the replacement of animal-derived antibodies. So we are constantly adding to this Ankyron catalogue, and we have several ongoing collaborations. So we do look forward to sharing more of this data in the future. But we are also looking to expand the catalogue in some very specific areas. So infectious diseases like hepatitis, Ebola, HIV, malaria, as well as cancer, neuroscience, and autoimmunity, just to name a few. So if you or any of your colleagues are interested in having some Ankyrons generated against a large array of proteins in these specific areas, or even if you just have a protein of interest and you simply don't have the tools at hand to visualize it, then please do get in touch and we'd be happy to sort of have those discussions with you. And we can also potentially generate these reagents for you completely free of charge as part of our privileged access program. And this is as we look to just broaden the scope of our catalogue offering. So with that, I'm all finished. Thank you for listening. And if we have time, I'd be happy to take any questions.
Great. Thank you, Charlie. I am going to read just a couple of quick questions from the chat, if that's okay.
Nice.
Yes, this is from Susan. It said, would it be possible to explain a little bit more about the labeling of peptide MHC complexes via Ankyrons? In the table, it seems as if they could also be used to find virus-specific T cells, just like traditional pentamers. Could you just explain a little bit about that slide?
Yes, so the Ankyrons that we generate are specific against the kind of peptide and MHC that would be presented on the surface of antigen-presenting cells, whereas our pentamers and our tetramers are, they bind to the specific populations of CD4 and CD8 cells themselves, so they don't recognize the MHC peptide complex on the surface. They recognize the CD8 and the CD4 cells themselves, so they do bind to different things.
Okay, thank you.
Great. And then there's also a question from Petcog. It says, can you give a clue how many Ankyron repeat domains make contact with the Ankyron epitope on the target material or antigen, or how many Ankyron repeats are in one reagent molecule?
I'm not sure. I'm also not sure if we could disclose that. That might be a Jeremy question.
Yeah, thanks, Charlie. Yeah, so ultimately, yeah, we can't disclose that specific information, but ultimately the way that the Ankyrons are generated is really through our randomized library, which obviously contains those repeats. So the Terra library of the trillion variants enables us to select those Ankyrons with exquisite specificity. So yeah, that specific information unfortunately isn't something that we can share.
Thank you.
Just a couple of quick ones. Disha, would you like to ask your question?
Yeah, so my question is it possible to generate Ankyron against HCPs, which are host cell proteins?
Sorry, could you repeat that? It was just a little quiet.
It is, can we generate Ankyrons against HCPs, which are host cell proteins? So we need to generate antibodies and show coverage, that it is covering all the host cell proteins. So is it possible with Ankyrons?
So when we generate our Ankyrons, correct me if I'm wrong, but we don't know exactly where on the protein it is binding to. So it could be to the protein as a whole, or it could be a specific domain of the protein.
So it would be population of protein, which will not include our product of interest. So apart from that, it will include all the proteins of null cell, and we need to show the coverage that antibodies generated against it is covering all the HCPs. So is it possible?
I can try and answer that as well, Disha. So yeah, the Ankyrons can certainly bind to host cell proteins, but we have to then use those host cell proteins obviously as the target to identify Ankyrons against. And you can't really do that with a very complex mixture. Usually you do need to use a defined specific target protein. So we would, you could generate a library of Ankyrons against individual host cell proteins that were specifically defined, but we need to have those defined HCPs in the first instance.
Okay, and in that case, regulatory agencies will be okay with that results, or they'll specifically ask for, I mean, that kind of results never submitted to regulatory agencies, or it will be through antibody coverage only?
Unknown. It's not been shown yet. So I don't know if that's the case with antibodies as well. I mean, if you need to generate an antibody against HCPs, it's going to be exactly the same principle. So yeah, I don't know, but it would be the same principle.
All right, thank you.
Okay.
You.
Let's just one last question before we move on from Subhankar. Would you like to ask your question?
Yeah, so thank you very much. It's a very nice presentation. So I have a question regarding how the way you actually select these Ankyrons. That's not a cell-based assay, if I understood correctly. Now, if we want to generate equivalent of like a blocking antibody for a receptor, so is that then comes to the users to screen from your library, or do you have any way to target these into a ligand binding pocket or something equivalent?
Yes, I think usually what we would do is we would generate and send you as many clones as we have as possible. And then that would be kind of down to you to test if they had any antagonistic or blocking capabilities or things like that. So that was kind of similar to what those collaborators did with the SARS-CoV-2 Ankyrons.
So in a very shortly, so in that case, you actually generate your Ankyrons based on the full length protein, or
it depends. We can do it to the whole protein, or if there's a specific domain of a protein that you do want to target, then we could just express that domain and then pick out any Ankyrons that are specific to that domain. We can do that. But in terms of actually then assessing if they have that capability, that would come down to you, I think.
all right. Okay. Thanks a lot. Thanks.
Thank you.
Great. Thanks, Charlie. And thanks everyone for those questions.